1.3 Physical Foundations. Living cells and organisms must perform work to stay alive and to reproduce themselves. The synthetic reactions that occur within cells, like the synthetic processes in any factory, require the input of energy. Energy input is also needed in the motion of a bacterium or an Olympic sprinter, in the flashing of a firefly or the electrical discharge of an eel. And the storage and expression of information require energy, without which structures rich in information inevitably become disordered and meaningless. In the course of evolution, cells have developed highly efficient mechanisms for coupling the energy obtained from sunlight or chemical fuels to the many energy-requiring processes they must carry out. One goal of biochemistry is to understand, in quantitative and chemical terms, the means by which energy is extracted, stored, and channeled into useful work in living cells. We can consider cellular energy conversions like all other energy conversions in the context of the laws of thermodynamics. Living organisms exist in a dynamic steady state, never at equilibrium with their surroundings. The molecules and ions contained within a living organism differ in kind and in concentration from those in the organism's surroundings. A paramecium in a pond, a shark in the ocean, a bacterium in the soil, an apple tree in an orchard all are different in composition from their surroundings, and, once they have reached maturity, maintain a more or less constant composition in the face of a constantly changing environment. Although the characteristic composition of an organism changes little through time, the population of molecules within the organism is far from static. Small molecules, macromolecules, and supramolecular complexes are continuously synthesized and broken down in chemical reactions that involve a constant flux of mass and energy through the system. The hemoglobin molecules carrying oxygen from your lungs to your brain at this moment were synthesized within the past month. By next month they will have been degraded and entirely replaced by new hemoglobin molecules. The glucose you ingested with your most recent meal is now circulating in your bloodstream. Before the day is over, these particular glucose molecules will have been converted into something else, carbon dioxide or fat, perhaps, and will have been replaced with a fresh supply of glucose, so that your blood glucose concentration is more or less constant over the whole day. The amounts of hemoglobin and glucose in the blood remain nearly constant because the rate of synthesis or intake of each just balances the rate of its breakdown, consumption, or conversion into some other product. The constancy of concentration is the result of a dynamic steady state, a steady state that is far from equilibrium. Maintaining this steady state requires the constant investment of energy. When a cell can no longer obtain energy, it dies and begins to decay toward equilibrium with its surroundings. We consider below exactly what is meant by steady state and equilibrium. Organisms transform energy and matter from their surroundings. For chemical reactions occurring in solution, we can define a system as all the constituent reactants and products, the solvent that contains them, and the immediate atmosphere, in short, everything within a defined region of space. The system and its surroundings together constitute the universe. If the system exchanges neither matter nor energy with its surroundings, it is said to be isolated. If the system exchanges energy, but not matter with its surroundings, it is a closed system. If it exchanges both energy and matter with its surroundings, it is an open system. A living organism is an open system. It exchanges both matter and energy with its surroundings. Organisms obtain energy from their surroundings in two ways. 1. They take up chemical fuels, such as glucose, from the environment and extract energy by oxidizing them. See box 1 to 3, case 2, or 2. They absorb energy from sunlight. The first law of thermodynamics describes the principle of the conservation of energy. In any physical or chemical change, the total amount of energy in the universe remains constant, although the form of the energy may change. This means that while energy is used by a system, it is not used up, rather, it is converted from one form into another, from potential energy in chemical bonds, say, into kinetic energy of heat and motion. Cells are consummate transducers of energy, capable of interconverting chemical, electromagnetic, mechanical, and osmotic energy with great efficiency. Fig. 1 to 26. Box 1. The Case 1. We know that Case 2. Entropy surround H2O are undergo an increase. C. We can the atoms can whenever case the there is a in addition to in this the flow of electrons provides energy for organisms. Nearly all living organisms derive their energy, directly or indirectly, from the radiant energy of sunlight. In the photoautotrophs, light-driven splitting of water during photosynthesis releases its electrons for the reduction of CO2 and the release of O2 into the atmosphere. $IMG ID equals IMG 0066 SRC equals /images /F0022-02.jpg roll equals presentation alt equals / Non-photosynthetic organisms, chemotrophs, obtain the energy they need by oxidizing the energy-rich products of photosynthesis stored in plants, then passing the electrons thus acquired to atmospheric O2 to form water, CO2, and other end products, which are recycled in the environment. C6H12O6 plus 6026 CO2 plus 6H2O plus energy yielding oxidation of glucose. Thus autotrophs and heterotrophs participate in global cycles of O2 and CO2, driven ultimately by sunlight, making these two large groups of organisms interdependent. Virtually all energy transductions in cells can be traced to this flow of electrons from one molecule to another, in a downhill flow from higher to lower electrochemical potential. As such, this is formally analogous to the flow of electrons in a battery-driven electric circuit. All these reactions involved in electron flow are oxidation-reduction reactions. One reactant is oxidized, loses electrons, as another is reduced, gains electrons. Creating and maintaining order requires work and energy. As we've noted, DNA, RNA, and proteins are informational macromolecules. The precise sequence of their monomeric subunits contains information, just as the letters in this sentence do. In addition to using chemical energy to form the covalent bonds between these subunits, the cell must invest energy to order the subunits in their correct sequence. It is extremely improbable that amino acids in a mixture would spontaneously condense into a single type of protein, with a unique sequence. This would represent increased order in a population of molecules, but according to the second law of thermodynamics, the tendency in nature is toward ever greater disorder in the universe. Randomness in the universe is constantly increasing. To bring about the synthesis of macromolecules from their monomeric units, free energy must be supplied to the system, in this case, the cell. We discussed the quantitative energetics of oxidation-reduction reactions in Chapter 13. $IMG ID equals IMG 0067 SRC equals /images /F0023-0 J. Key convention: the randomness or disorder of the components of a chemical system is expressed as entropy S, box 1 to 3. We will give a more rigorous definition of entropy in Chapter 13. Any change in randomness of the system is expressed as entropy change, delta S, which by convention has a positive value when randomness increases. J. Willard Gibbs, who developed the theory of energy changes during chemical reactions, showed that the free energy content, G, of any closed system can be defined in terms of three quantities: enthalpy H, reflecting the number and kinds of bonds, entropy S, and the absolute temperature T in Kelvin. The definition of free energy is G equals HTS. When a chemical reaction occurs at constant temperature, the free energy change, delta G, is determined by the enthalpy change, delta H, reflecting the kinds and numbers of chemical bonds and non-covalent interactions broken and formed, and the entropy change, delta S, describing the change in the system's randomness. Where, by definition, delta H is negative for a reaction that releases heat, and delta S is positive for a reaction that increases the system's randomness. A process tends to occur spontaneously only if delta G is negative, if free energy is released in the process. Yet cell function depends largely on molecules, such as proteins and nucleic acids, for which the free energy of formation is positive. The molecules are less stable and more highly ordered than a mixture of their monomeric components. To carry out these thermodynamically unfavorable, energy-requiring, exergonic reactions, cells couple them to other reactions that liberate free energy, exergonic reactions, so that the overall process is exergonic. The sum of the free energy changes is negative. $IMG SRC equals /images /F0024-01.jpg area described B equals Longerelch 1. Figure. The usual source of free energy in coupled biological reactions is the energy released by breakage of phosphoanhydride bonds, such as those in adenosine triphosphate, ATP Fig. 1-27, and guanosine triphosphate, GTP. Here, each P represents a phosphoryl group. $IMG ID equals IMG 0068 SRC equals /images /F0024-02.jpg role equals presentation alt equals /. When these reactions are coupled, the sum of delta G1 and delta G2 is negative. The overall process is exergonic. By this coupling strategy, cells are able to synthesize, and maintain the information-rich polymers essential to life. Energy coupling links reactions in biology. The central issue in bioenergetics, the study of energy transformations in living systems, is the means by which energy from fuel metabolism or light capture is coupled to a cell's energy-requiring reactions. In thinking about energy coupling, it is useful to consider a simple mechanical example, as shown in Figure 1-28a. An object at the top of an inclined plane has a certain amount of potential energy as a result of its elevation. It tends to slide down the plane, losing its potential energy of position as it approaches the ground. When an appropriate string and pulley device couples the falling object to another, smaller object, the spontaneous downward motion of the larger can lift the smaller, accomplishing a certain amount of work. The amount of energy available to do work is the free energy change, delta G. This is always somewhat less than the theoretical amount of energy released, because some energy is dissipated as the heat of friction. The greater the elevation of the larger object, the greater the energy released, delta G, as the object slides downward, and the greater the amount of work that can be accomplished. The larger object can lift the smaller only because, at the outset, the larger object was far from its equilibrium position. It had at some earlier point been elevated above the ground, in a process that itself required the input of energy. $IMG SRC equals /images /F0024-03.jpg aria described B equals longerulch 1_27 alt equals several chemical structure Figure Figure 1. How does this apply in chemical reactions? In closed systems, chemical reactions proceed spontaneously until equilibrium is reached. When a system is at equilibrium, the rate of product formation exactly equals the rate at which product is converted to reactant. Thus there is no net change in the concentration of reactants and products. The energy change as the system moves from its initial state to equilibrium, with no changes in temperature or pressure, is given by the free energy change, delta G. The magnitude of delta G depends on the particular chemical reaction, and on how far from equilibrium the system is initially. Each compound involved in a chemical reaction contains a certain amount of potential energy, related to the kind and number of its bonds. In reactions that occur spontaneously, the products have less free energy than the reactants, and thus the reaction releases free energy, which is then available to do work. Such reactions are exergonic, the decline in free energy from reactants to products is expressed as a negative value. Endergonic reactions require an input of energy, and their delta G values are positive. As in mechanical processes, only part of the energy released in exergonic chemical reactions can be used to accomplish work. In living systems, some energy is dissipated as heat or lost to increasing entropy. Keq and delta G degree are measures of a reaction's tendency to proceed spontaneously. The tendency of a chemical reaction to go to completion can be expressed as an equilibrium constant. For the reaction in which a moles of A react with B moles of B to give C moles of C and D moles of D, alpha A plus BBCC plus DD. The equilibrium constant, Keck, is given by where A, EQ is the concentration of A, B, EQ the concentration of B, and so on, when the system has reached equilibrium. Keck is dimensionless, that is, has no units of measurement, but as we explain on page 59, we will include molar units in our calculations to reinforce the point that molar concentrations, represented by the square brackets, must be used in calculating equilibrium constants. A large value of Keck means the reaction tends to proceed until the reactants are almost completely converted into the products. Worked example 1-to-1 or equilibrium in cells. The equilibrium constant, Keck, for if the measured cellular concentrations are ATP equals 5 mm, ADP equals 0.5 mm, and pi equals 5 mm, is this reaction at solution? The definition of the Keck equals ADP pi from the measured cellular concentration. This value worked reaction in cell for the react glucose plus the equal mmAd Keck equals 7.8 times 10 in living mm/. Gibbs showed that G, the actual free energy change for any chemical reaction, is a function of the standard free energy change, G degree, a constant that is characteristic of each specific reaction and a term that expresses the initial concentrations of reactants and products. Where I is the initial concentration of A and so forth, R is the gas constant, and T is the absolute temperature. G is a measure of the distance of a system from its equilibrium position. When a reaction has reached equilibrium, no driving force remains, and it can do no work. G equals 0. For this special case, AI equals AEQ and so on for all reactants and products, and substituting 0 for G in Keck for an equation 1-to-1, we obtain the relationship G degree, from which we see that G degree is simply a second way, besides Keck, of expressing the driving force on a reaction. Because Keck is experimentally measurable, we have a way of determining G degree, the thermodynamic constant characteristic of each reaction. The units of G degree and G are joules per mole, or calories per mole. When Keck 1, G degree is large and negative. When Keck 1, G degree is large and positive. From a table of experimentally determined values of either Keck or G degree, we can see at a glance which reactions tend to go to completion and which do not. One caution about the interpretation of G degree: thermodynamic constants such as this show where the final equilibrium for a reaction lies, but tell us nothing about how fast that equilibrium will be achieved. The rates of reactions are governed by the parameters of kinetics, a topic we consider in detail in Chapter 6. In biological organisms, just as in the mechanical example in Figure 128a, an exergonic reaction can be coupled to an endergonic reaction to drive otherwise unfavorable reactions. Figure 1-28b, a type of graph called a reaction coordinate diagram, illustrates this principle for the conversion of glucose to glucose 6-phosphate, the first step in the pathway for oxidation of glucose. The simplest way to produce glucose 6-phosphate would be Reaction 1, glucose plus pi glucose 6-phosphate, endergonic, G1 is positive. Don't be concerned about the structures of these compounds now, we describe them in detail later in the book. This reaction does not occur spontaneously, G1 is positive. A second, highly exergonic reaction can occur in all cells. Reaction 2, ATP-ADP plus pi, exergonic, G2 is negative. These two chemical reactions share a common intermediate, pi, which is consumed in reaction 1 and produced in reaction 2. The two reactions can therefore be coupled in the form of a third reaction, which we can write as the sum of reactions 1 and 2, with the common intermediate, pi, omitted from both sides of the equation. Reaction 3, glucose plus ATP glucose 6-phosphate plus ADP. Because more energy is released in reaction 2 than is consumed in reaction 1, the free energy change for reaction 3, G3, is negative, and the synthesis of glucose 6-phosphate can therefore occur by reaction 3. Worked changes given solution 1, 2, the cup group from worked synthesis. If the equal AT solution, this is the worked change glucose. What is the glucose solution equation? GD. Notice, enzymes promote sequences of chemical reactions. All biological macromolecules are much less thermodynamically stable than their monomeric subunits, yet they are kinetically stable. Their uncatalyzed breakdown occurs so slowly, over years rather than seconds, that, on a time scale that matters for the organism, these molecules are stable. Virtually every chemical reaction in a cell occurs at a significant rate only because of the presence of enzymes biocatalysts that, like all other catalysts, greatly enhance the rate of specific chemical reactions without being consumed in the process. Figure. The path from reactants to products almost invariably involves an energy barrier, called the activation barrier, Fig. 1 to 29, that must be surmounted for any reaction to proceed. The breaking of existing bonds and formation of new ones generally requires, first, a distortion of the existing bonds to create a transition state of higher free energy than either reactant or product. The highest point in the reaction coordinate diagram represents the transition state, and the difference in energy between the reactant in its ground state and in its transition state is the activation energy G. An enzyme catalyzes a reaction by providing a more comfortable fit for the transition state, a surface that complements the transition state in stereochemistry, polarity, and charge. The binding of enzyme to the transition state is exergonic, and the energy released by this binding reduces the activation energy for the reaction and greatly increases the reaction rate. A further contribution to catalysis occurs when two or more reactants bind to the enzyme's surface close to each other, and with stereospecific orientations that favor the reaction. This increases by orders of magnitude the probability of productive collisions between reactants. As a result of these factors and several others, discussed in Chapter 6, Enzyme-Catalyzed, Reactions commonly proceed at rates greater than 10-12 times faster than the uncatalyzed reactions. That is a million times faster. Cellular catalysts are, with some notable exceptions, proteins. Some RNA molecules have enzymatic activity, as discussed in chapters 26 and 27. Again, with a few exceptions, each enzyme catalyzes a specific reaction, and each reaction in a cell is catalyzed by a different enzyme. Thousands of different enzymes are therefore required by each cell. The multiplicity of enzymes, their specificity, the ability to discriminate between reactants, and their susceptibility to regulation give cells the capacity to lower activation barriers selectively. This selectivity is crucial for the effective regulation of cellular processes. By allowing specific reactions to proceed at significant rates at particular times, enzymes determine how matter and energy are channeled into cellular activities. The thousands of enzyme-catalyzed chemical reactions in cells are functionally organized into many sequences of consecutive reactions, called pathways, in which the product of one reaction becomes the reactant in the next. Some pathways degrade organic nutrients into simple end products in order to extract chemical energy and convert it into a form useful to the cell. Together, these degradative, free energy-yielding reactions are designated catabolism. The energy released by catabolic reactions drives the synthesis of ATP. As a result, the cellular concentration of ATP is held far above its equilibrium concentration, so that G for ATP breakdown is large and negative. Similarly, catabolism results in the production of the reduced electron carriers NADH and NADPH, both of which can donate electrons in processes that generate ATP or drive reductive steps in biosynthetic pathways. Other pathways start with small precursor molecules and convert them to progressively larger and more complex molecules, including proteins and nucleic acids. Such synthetic pathways, which invariably require the input of energy, are collectively designated anabolism. The overall network of enzyme-catalyzed pathways, both catabolic and anabolic, constitutes cellular metabolism. ATP and the energetically equivalent nucleoside triphosphate cytidine triphosphate, CTP, uridine triphosphate, UTP, and guanosine triphosphate, GTP, is the connecting link between the catabolic and anabolic components of this network, shown schematically in Fig. 1-30. The pathways of enzyme-catalyzed reactions that act on the main constituents of cells' proteins, fats, sugars, and nucleic acids are virtually identical in all living organisms. Describe Figure 1. Metabolism is regulated to achieve balance and economy. Not only do living cells simultaneously synthesize thousands of different kinds of carbohydrate, fat, protein, and nucleic acid molecules and their simpler subunits, but they do so in the precise proportions required by the cell under any given circumstance. For example, during rapid cell growth, the precursors of proteins and nucleic acids must be made in large quantities, whereas in non-growing cells the requirement for these precursors is much reduced. Key enzymes in each metabolic pathway are regulated so that each type of precursor molecule is produced in a quantity appropriate to the current requirements of the cell. Consider the pathway in E. coli that leads to the synthesis of the amino acid isoleucine, a constituent of proteins. The pathway has 5 steps catalyzed by 5 different enzymes. A through F represent the intermediates in the pathway. $IMG ID equals IMG 0. If a cell begins to produce more isoleucine than it needs for protein synthesis, the unused isoleucine accumulates and the increased concentration inhibits the catalytic activity of the first enzyme in the pathway, immediately slowing the production of isoleucine. Such feedback inhibition keeps the production and utilization of each metabolic intermediate in balance. Throughout the book, we use $IMG ID equals IMG 0069 SRC equals. Summary 1.3 Physical Foundations. Although the concept of discrete pathways is an important tool for organizing our understanding of metabolism, it is an oversimplification. There are thousands of metabolic intermediates in a cell, many of which are part of more than one pathway. Metabolism would be better represented as a web of interconnected and interdependent pathways. A change in the concentration of any one metabolite would start a ripple effect, influencing the flow of materials through other pathways. The task of understanding these complex interactions among intermediates and pathways in quantitative terms is daunting, but systems biology, discussed in Chapter 15, has begun to offer important insights into the overall regulation of metabolism. Cells also regulate the synthesis of their own catalysts, the enzymes, in response to increased or diminished need for a metabolic product. This is the substance of Chapter 28. The regulated expression of genes, the translation from information in DNA to active protein in the cell and synthesis of enzymes or other layers of metabolic control in the cell. All layers must be taken into account when describing the overall control of cellular metabolism. Summary 1.3 Physical Foundations. Living cells are open systems, exchanging matter and energy with their surroundings, extracting and channeling energy to maintain themselves in a dynamic steady state distant from equilibrium. Energy is obtained from sunlight or chemical fuels by converting the energy from electron flow into the chemical bonds of ATP. The tendency for a chemical reaction to proceed toward equilibrium can be expressed as the free energy change, G, which has two components, enthalpy change, H, and entropy change, S. These variables are related by the equation G equals HTS. When G of a reaction is negative, the reaction is exergonic and tends to go toward completion. When G is positive, the reaction is endergonic and tends to go in the reverse direction. When two reactions can be summed to yield a third reaction, the G for this overall reaction is the sum of the G values for the two separate reactions. The reactions converting ATP to pi and ADP, or to AMP and PPI, are highly exergonic, large negative G. Many endergonic cellular reactions are driven by coupling them, through a common intermediate, to these highly exergonic reactions. The standard free energy change for a reaction, G degree, is a physical constant that is related to the equilibrium constant by the equation g degree equals RT ln kek. Most cellular reactions proceed at useful rates only because enzymes are present to catalyze them. Enzymes act in part by stabilizing the transition state, reducing the activation energy, G, and increasing the reaction rate by many orders of magnitude. The catalytic activity of enzymes in cells is regulated. Metabolism is the sum of many interconnected reaction sequences that interconvert cellular metabolites. Each sequence is regulated to provide what the cell needs at a given time, and to expend energy only when necessary.